Teacher from Northeastern Ontario and a long-time author with Nelson Education, and so I want to do a little talk today on just kind of where you know, some of the things that are happening in genetics, and then hopefully have an opportunity to have you guys kind of discuss what the implications are for how we teach genetics and what we choose to teach and sort of time allotments and that sort of thing in genetics. So I want to start out though by maybe Jeff will just give us just a little introduction to a new Nelson initiative.
We should point out, first of all, you may appear on film today. So
oh yeah.
If you have some issues with that, please let us know. But we're going to be filming, we filmed this one, but some of our other sessions. If you go to weinspirefutures.com, you will be able to continue this conversation that you have today online. So you'll find videos from this session and others. Doug will be able to interact with you after today. Yep. If you go on there, and there are places there for you to leave your stories about your classroom and the genetics and whatever else other things you're doing in your classroom. So basically it's trying to build a community of educators where you can continue to share this great stuff that happens here, but to do it online. So if you come over to the booth, we're just at the end of the hallway, right at the end, and you can see what that's all about. But it's a way to continue asking Doug questions. I know at the end of every session, we've still got people beating down the door when you're trying to get the next session started. So he'll be able to interact with you still and be able to hear what you're doing in your classroom, plus maybe share some insights that he has. So we really encourage you to come and have a look at that site.
Okay. Thanks, Jeff. Yeah, and yeah, it's you know, an educational community, a place to go and post you know, blogs. There's Pinterest boards there. I'm not a pinner yet. I guess I should start pinning. But you know, interesting websites related to science and education and all that sort of stuff. My first blog entry was about a fantastic trip I took this summer to Panama with students and kind of how that can inspire students and teachers. And so if anyone's interested in learning about how to take tropical adventures with myself or students, they can ask me questions. So those opportunities. Okay, so you know, genetics is a very, very high-tech kind of a modern thing. So of course there are, you know, apps for your cell phone. Maybe some of you are using this now. I don't know. This is you know, an app for figuring out what good restriction enzymes to use for various digests in your little research lab. And so there's an enormous amount of data in genetics, right? Compiling huge amounts of data, and therefore we need lots of ways of accessing and working with that data to our advantage. So this is one that does that sort of stuff. Then very oddly yesterday, because I'm not a geneticist, I got an email asking me if I was, you know, interested in signing on to GQIP, which is Genome Quest's. It's like a piece of software. It only costs $3,500. It's a one-time thing. It's a deal now. If you're a first-time member, you can get in on the ground floor, $3,500. And the only reason I shared this with you, well, it was sort of odd that it was this genetics technology offer in my mailbox, but I just, you know, I clicked on the little 3-minute tutorial, and it kind of shows you the scope of where they're at. All this thing is used for is if you're a pharmaceutical company, a research lab, a Monsanto, like agro-business, you literally take a little piece of genetic code, right? A little string of base pairs or a little amino acid string. You put it into the search engine, and it searches the world's databases for where that shows up in relation to patents. So it's trying to find out, okay, so I'm, you know, working on this little, you know, bit of biology, and I discovered this little sequence. I wonder, I suppose, if anyone else has patented it. What stage of patent they're in? Like, what are all of the implications? And when you enter a little thing, you get like this massive number of hits from around the world, and you get your patent lawyers and everything else. But anyways, the level of sophistication is really quite dramatic, and the scale that this is being done on. You know, it's not like a couple of companies are out there trying to patent a couple of pieces of DNA. it's a massive scale that this is being done on. Oh, then there's good news. In genetics applications, you know, they've discovered the foaming gene. So, you know, you get those really nice foams on some of those English beers. Well, they've discovered the gene that's responsible for the protein that, you know, influences foaming. So there's well, and of course, this is big business, right? Yeast is the best known eukaryotic cell on the planet for good reason, because yeast in brewing and I suppose baking and you know, fermenting wines is like a huge money-making organism, and so it's extremely well known. But anyway, then there's all kinds of interesting trivia about what we find out about the genomes of organisms that we investigate. So here's a weird one. You know, rotifers, one of the coolest things that you show students in pond water under the microscope. These crazy little animals, very, very small animals, so they're eukaryotes. This one, they don't have sex, and so, you know, the students would kind of think that was not very interesting, but they have accumulated like 10% of their functioning genes seem to be genes that they've picked up from just eating, kind of like, they kind of feed on detritus. You know, they're taking in little you know, bacteria, little single-celled algae and fungi, and they have over their, you know, 80 million years or so, they've accumulated and are using these other genes. One of the things that they use these genes for in this very, very odd bit of biology is some of these guys, well in particular this one, they can dry out, so they live in little pond water, and the pond can dry out and they can be completely desiccated and then just sit there for years. And then, you know, with a drop of water, they, you know, rehydrate and come back to life. And some of the genes that allow them to do that have been pirated from microbes. So a strange little thing. Here's an example of one of, you know, many, many genetic discoveries. You know, the human brain, one of the reasons that we have this amazing cognitive ability is because we have a very large surface area of our cerebral cortex. And in some individuals with these rare disorders, they have a smooth part of their brain, no infolding. So it gives them all kinds of, you know, mental handicaps. But you know, they've discovered that strange disease is associated with a two base pair deletion in, you know, in a particular gene. So not only does it give you insight into that medical problem, but it sort of gives you insight into the evolution of higher, you know, complex brains, because whatever's needed to start convoluting our brains was very, very significant in the evolution of mammals and in particular primates. Huntington's disease, one that we often use in high school, you know, we use it in our grade 11 biology text as a good example of a dominant recessive gene, and I won't get into all of the details about it, but it's a very, very interesting gene and disease in many, many ways, and you know, it plays out in influencing the mitochondria, but there's just all kinds of things happening that, you know, that are promising, right? So they've got this antioxidant, this strange synthetic antioxidant that they've been using that seems to protect mitochondria from damage and the DNA within mitochondria from damage. And so in a mouse model, and they genetically engineered the mouse to get Huntington's disease, so you have to do that first, and then you play around with possible treatments. So that's been promising. These are all very new things. This is all within the last year or less. And then this new technology of using these, what they call zinc finger proteins, which can go in and bind to particular sequences of DNA and then kind of shut down that DNA by sort of blocking it from being transcribed. And so there's been some promising work in, again, in mouse models with that strategy to prevent Huntington's from manifesting itself. So a lot, you know, a ways away from human trials, but very promising because of our understanding of genetics. Also now, in an evolutionary sense, you know, it's unusual to, you know, to have, you know, genetic diseases that are very, very disadvantageous, you know, tend to get weeded out through natural selection. But then, you know, there's always the question of, are there benefits to some of these genes? So a recent study has shown that people who have the Huntington gene actually are better learners. So, you know, maybe one of the reasons that that gene sort of hung on in human evolution was because it gave you an advantage when you were young. The fact that it gives you a disadvantage when you're old isn't all that important, really, in humans, because you're mostly stopped reproducing, although there's also this thing called the grandmother hypothesis, but that's another story, next talk, after this one on evolution. But anyway, so there may actually be advantages to something as serious a disease as Huntington's. So I wanted to just kind of go through these topics and address them a little bit, some a little quicker than others, but we have moved a long way since Mendel, and in fact the last discussion question for the group, in a sense, will be how much should we be focusing in high school biology on the science of genetics, the Mendel stuff, and how much on the you know, the STSC issue stuff. So, you know, in the year 2000, that was a big year for genetics, and the draft publication of the Human Genome Project and the Venter Celera draft as well. Then we have these new topics, epigenetics and pharmacogenomics, which are, you know, well, kind of new because we're learning more. Direct-to-consumer genetic tests. Christmas is coming up, so I want to mention that. And the current status of gene therapy, and then sort of these last questions about teaching kids. So you know, this is one of the very first genomes ever sequenced. This is a very, very small bacterium. It's actually infectious, well, it lives in the urinary tracts of humans. It's an infectious little bacteria, but only has 580,073 base pairs about. It's got only about 500,000 base pairs, and so it's one of the smallest genomes, and so was sequenced first. One of the I think the second genome to be sequenced, and this was the first map that came out. I think this was in like 1995. And it was available to download. I downloaded the genome and printed it out, like the whole ATCCG, 580,000, and just put it in a binder and brought it to school and said, you know, there it is. Like there's the entire genetic code for a living thing. It's sort of bizarre to be able to hold that in your hand, like this monumental breakthrough in biology and our understanding. And you know, there was lots of cool things you could do in your classes with these maps, because they're all color-coded, meaning certain types of you know, gene function categories and that sort of thing. So what have we done now, though? You know, mycoplasma, I was all excited about that one, the Human Genome Project. Well, that's kind of important because that's like me and my grandmother, you know. But now we've sequenced 3,791 species are either completely sequenced or like sort of the final draft version is done. Okay, so of those, 172 are archaea. Not surprisingly, the vast majority are eubacteria, partly because they're a lot easier to sequence because they have so much smaller genomes, but we're also really, really interested in a lot of them because they cause us trouble. Right? There's a lot of disease organisms that are bacteria that we're very, very interested in. And then 182 eukaryotes, which is a lot. You know, there's, well, you know, obviously, you know, humans and yeast, and all kinds of important organisms in agriculture. You know, most of the world's major plants, agricultural plants have been sequenced, and livestock has been sequenced. The Anopheles mosquito, because it carries malarial organism, Plasmodium, has been sequenced. All of those kinds of things. So there's an enormous amount of sequence that's, and of course our closest relatives, chimpanzees and the bonobos and the orangutans and gorillas have all been sequenced. And there's about 15,000 that are like, you know, in stages of being done. So now, this is now routine. Okay, so 10 years ago this was still cutting edge, like wow, we've been able to do this. Now it's like, you know, did we get the funding to sequence that genome yet? Because I was hoping to do that, you know, like, you know, in my second semester as my thesis project or something. It's sort of that level. So here's an example of where they, this project was to sequence now this is to sequence in like 1,000 entire genomes of people from various, I don't want to call them races because we talked about that, where there are no human races, but different groups of people to try and look at human genetic diversity. So it took a couple of hundred scientists, 1,000 genomes and 14 ethnic groups. It's ultimately to be expanded to 2,500 genomes in this particular project. So they call it a deep catalog of human genetic variation. This is not available yet, so maybe next year, but there are many companies that have a technology that are close to this sort of target of the $1,000 genome. The idea that we're going to be able to sequence a genome for $1,000 in a day. And that is close, right? So again, now that took, the Human Genome Project took, well, let's just say close to 10 years. It took thousands of scientists, and it cost, I'm not sure enough, it was either $10 million or $100 million. Doesn't really matter. It was a lot of money, right? So now, and it's probably kind of inaccurate, I mean, it's like a draft version, right? So now for so there we go, savings of $10 million. So you're going to be able to do this for $1,000, like sort of in an afternoon. We have these other banks of genetic information, like these expressed sequence tags and these other bits of genetic information that are kind of like the important markers that aren't the whole genome. But here we have a project where in California, this is the largest project in the United States, I'm not sure about in the world, but they have a database now for 100,000 Californians, and they're using this, to look at relationships between various aspects of the genome and diseases, human physical characteristics, that kind of thing. They found out more things about telomeres and aging with this study, and I'll talk a little bit about that. The UK working on 500,000, and these are the kinds of storage libraries, all automated, robotic little storage facilities for holding all of this information and samples and that sort of thing. We've also now, of course, we can even look at genomes of things that, well,